All right, let us continue as we move forward in our analysis of Chapter 6. We're going to now finish the chapter out with Part 2. We're looking at, of course, microbial nutrition and growth. And last we spoke, we were talking about osmosis. And again, that's the movement of solutions or materials, water, through a membrane, a selectively permeable membrane, so that the solute, if you look in the over the red, they can't fit through the pores where the water can. And it's really all osmosis is about is how much the red little beads here are displacing the water. And so if it's displacing a certain amount of water, the water now has to compensate and add into it. And so if there's more of these red beads, let's say on the outside, then whatever is separated from that is going to lose water to try to compensate for the amount of water on either side of that membrane. So that's the Dr. V view of osmosis. But let's look at this. So, as I mentioned, in an isotonic solution, life is good. The amount of water inside and outside of the cell are the same. So there is no real net movement. Now, it is moving back and forth, just normal equal balance there, equivalent type of movement, but hypotonic, hypo meaning like hypodermic, goes below. The net diffusion of water is into the cell, thus swells the protoplasm. What that means is there's a fair amount of displacement. If you notice that there's several of those little teeny of the blue balls here inside and few on the outside. So what's going to try to occur here is to get an equal balance of the water pressure, the same amount of water and spacing between the two, and eventually the cell will burst. And that is a hypotonic solution that it's in, so it's taking in consideration its environment, where a hypertonic or above is different. In this case, water diffuses out of the cell and shrinks the cytoplasmic membrane away from the cytowall. It's trying to fuse water into it, into the outside environment, to make the balance equal as best it can. But in the process, it loses all of its water and shrivels, and that could be really problematic. And cell will die. Okay, let's shift gears and talk about transport. In transport, we have two types. We have passive and active. Passive is just simple diffusion. It's just a fundamental property, Brownian movement. Things are shaking and moving, and as a result, molecules will move randomly around, and it's just passive. Facilitated diffusion is a molecules bind to a particular receptor in the membrane. So these are already made, no energy is required other than the fact that that protein that's sitting in the membrane had to be manufactured. But that's picky uni detail, but it did take energy to make that protein. But no more energy is required after it's been built or made. Active, on the other hand, requires adenosine triphosphate, the chemical bond energy in that last phosphate bond, to change the shape, thus facilitating the movement of whatever the substrate it wants to move. So in passive, we see that glucose binds into the protein. Now there's no energy added to this. It's just because of the chemical fit, and once the glucose, in this case, becomes in the active area or the active site of that protein, because of its charge, facilitates a net movement of that particular protein to kind of squeeze it, so the glucose comes inside the cell. And then because the charge of the glucose is now removed from that pocket, it now snaps back to its original position. It's very clever. This is how we facilitate very specific things across the membrane, like glucose. So the glucose molecule has that chemical fingerprint, or that hand and glove type of scenario. Once it's in the pocket, it makes that change, and that is passive. It doesn't require energy other than the fact to make the protein at the first place. Now, if you look at active is a different deal. That requires ATP. So if we have a particular protein, ATP binds to it, thus transferring the chemical bond energy, as you see here, making ADP. It now changes the characteristics of that protein, so it now flips and dumps whatever was loaded into that particular protein, moves it in, and that is through active process. In other words, the ATP actually changed the conformation of that protein shape. When it does change, it loses that chemical bond energy and snaps back to its original position. So you can see it's in this position, ATP binds, snaps, we move whatever the molecules that were loaded, and then ATP or ATP is broken away in the form of ADP, and we see the protein now snapping back to its original form. And that's active, and you can just remember active and ATP, both of course starting with that A. So active transport is a transport of nutrients against diffusion gradient, or in the same direction as the natural gradient, but at a much faster rate. The presence of specific membrane proteins, permeases and pumps, are there that facilitate and react to the presence of ATP, chemical bond energy, and in the form of that phosphate bond. Pretty straightforward. Endocytosis, eating and drinking by cells. So endocytosis, process of bringing things into the cell. So the cell encloses a substance into its membrane, simultaneously forming a vacuole, or sort of an enclosed area, and engulfs the substance. Now we have phagocytosis, which is the process of a cell eating, accomplished by amoebas and white blood cells, where they ingest matter. Pino, pen in Latin, in the conjunction form means drink. So if you want a pint of beer, you think of it that way, pinocytosis. So it's the process of a cell drinking, and by definition that's Latin's so nice for that. It gives you exactly what's going on. Alright, we're going to talk about 5 terms to express temperature-related growth and capabilities of microbes. And this is really straightforward. It's vocabulary, so just list those and let's talk about it. So there's certain maximum and minimum temperatures that things grow at for a particular species, and we group them into that. We know that they have minimum and maximum temperatures, and then there's an optimum, in other words, where they really like it. And so we're going to talk about the nomenclature, or the vocabulary. Psychrophiles, psychro is cold in Greek, and philes meaning liking. So the optimum temperature below 15 degrees, capable of growth at 0. So if you think you put things in the refrigerator and you're hoping that things just don't grow, well, not particularly true. Things like to grow at that temperature and will. And sometimes if you leave things in there too long, you notice that things are starting to wave at you as you open the door. That's what I'm talking about. So those are saprophilic types of organisms. Obligate with respect to cold and cannot grow above 20, so we don't have to worry about them as pathogens, but they may be preforming toxins which will survive and you eat it, and then you're going to be really sorry later. Natural inhabitants of saprophilic bacteria, fungi, algae or lakes, rivers, snowfields, polar ice, deep oceans, sort of rarely pathogenic, so I don't really worry about them. But it's a term we need to know. Psychotrophs, now that just means that they will grow, slow growing. In other words, they grow slowly in the cold but have an optimum temperature between 15 and 30. And this is damning from the standpoint that if you've ever had tainted sushi, which I have, Listeria monocytogenes is able to grow in the refrigerator and cause foodborne illness as staph. Now obviously it's not every 20 minutes, the doubling time, it's slower than that, but still it's actively growing and producing toxins and going to make the 4 hours after you eat it very challenging, let's put it that way. Middle phials, majority of medically significant ones, these grow in within the range of 20 to 40 degrees C, and that's, we look at 37 degrees as sort of our optimum for our body temperature. So we worry about these, and they inhabit animals and plants and soil in temperate areas, subtropical and tropical regions, which all of this is just common sense, but really don't let all the other distractions of the discussion. Mesophils is just a vocabulary word in the middle. Thermoduric microbes. Now the word duric means enduring, hard enduring. In other words, it can handle the heat, can survive short exposures to high temperatures, but are normally mesophils, so the higher temperatures may not kill it, and it comes back and grows. Common contaminated of heated or pasteurized foods. A lot of the spore formers we worry about are Bacillus and Clostridium species. Bacillus anthracis, anthrax, Clostridium difficile, C. diff, or Clostridium species that we're going to talk about in the lab in the anaerobe section. Thermophiles, which like the temperatures quite warm, live in soil and water associated with volcanic activity, compost piles. Trucks that go down the highway, the load of whatever they're carrying starts to steam, and that's because if you actually put your hand on a pile of, let's say, peat moss or something, it's very hot, and it's due to microorganisms growing, and the heat within that pack is much higher. So most eukaryotic forms cannot survive above 60. Extreme thermophiles can grow 80 and 121 degrees, like Thermophilus aquans, the strain that they use for PCR or polymerase chain reaction, grows at those high temperatures, and we take advantage of the DNA polymerase that can withstand higher temperatures. But as far as pathogens, we don't worry about the thermophiles. So you can see, it's a summary, all of the different types, and it's a good way to kind of look at it. Color is a very powerful memory tool, and cold being blue here is a saccharophile, and then we move to psychotrophs, and then the mesophiles, the thermophiles, and then the extreme thermophiles. And you can see the optimum is the highest part of the peaks at the temperatures that they prefer. Straightforward vocabulary. And gases, atmospheric gases that influence microbial growth are oxygen and CO2. O2 has the greatest impact on microbial growth. It's an important respiratory gas and a powerful oxidizing agent. In fact, blood and oxygen, blood is toxic, and it doesn't make sense, but it is, and it's because it's carrying oxygen, the heme does, and that's a strong oxidant, and it can damage tissues as typical. Microbes fall into one of three categories: those that use oxygen and detoxify it, those that can neither use oxygen nor detoxify it, and those that do not use oxygen but can detoxify it. So you can see that the three classes are pretty distinct. Oxygen enters the cellular reactions. It's transformed into several toxic products. Single oxygen, it's extremely reactive. It's looking for something to fill its outer vacancies to get the octet rule satisfied. Superoxide ions O2, which it's an ion, so you see the negative charge there. It's highly reactive. In fact, there's certain enzymes that bacteria have called superoxide dismutase that deals with this particular ion specifically. Hydrogen peroxide, which we have used in the lab, you see things bubble or not. If you remember, staph has an enzyme that breaks it down, catalase, and strep doesn't. But it's hydrogen peroxide, toxic to cells and used as a disinfectant. And you know that, you can buy that. It's usually in a brown bottle because it's so unstable that we try to filter the direct sunlight out of it by storing it in a dark type of bottle like we see. A hydroxyl radical is also highly reactive, and we know that's true from pH discussions that we've had. So most cells have developed enzymes that scavenge and neutralize these reactive oxygen byproducts. The two-step process requires two enzymes, superoxide dismutase or catalase. Now we just talked about both of those, but the superoxide ion is converted into hydrogen peroxide. So you can see that superoxide dismutase turns the highly reactive, the 2O2, into H2O2, which is hydrogen peroxide, and catalase will take that even further and break it into water and just regular oxygen, atmospheric oxygen, which can be diffused, so it's not harmful to the cell. And this is why they talk about antioxidants and things in your diet and those sorts of things that deal with these oxygen radicals. If you don't believe that blood and oxygen is toxic, all you have to do is, with permission of course, ask someone that's elderly sitting in the chair. They'll usually have their shin exposed and it'll be very warm. And so if you were to touch it, you notice it's quite hot. Blood stasis is a problem. The best cure for that is to get up and move around. But when you have, when you're older, of course, as I'm finding out, things start to hurt a lot more and you don't get around as much. But the best thing to do is to move around and get that blood to move. But that thickening of the cell and of the cells along that shin, and you notice it is reddened, and it actually thickens the skin, and it's a problem. So you've got to deal with it. You'll see it. Aerobes can use gaseous oxygen in their metabolism and possess enzymes needed to process toxic products. Microaerophiles are harmed by normal atmospheric concentrations of oxygen but require a small amount in its metabolism. So these not directly exposed. So Borrelia burgdorferi, the causative agent for Lyme disease, likes to go intracellular into our cells. And in poorly vascularized regions, now you know why. They're microaerophiles and they're harmed by the atmospheric concentrations. They actually detect the host by, they have ways of detecting CO2. They are not O2 type of organisms. They actually prefer the CO2 on the side, but they still have to have oxygen for its metabolism. Helicobacter pylori causes the ulcers, and it's in a very low pH environment, and again, it's a microaerophile. Facultative anaerobes do not require oxygen for metabolism, but use it when it's present. A lot of gram-negative intestinal bacteria like E. coli will do that, and Staphylococcus will do that. And so in a tube, we're going to be talking about growth in a tube because it's typically liquid growth, and we can facilitate oxygen diffusion more easily through a liquid. Anaerobes lack the metabolic enzyme system for using oxygen in respiration. We'll get anaerobes still lack enzymes and die in its presence. And I used to work with anaerobes a lot, and we've had a situation where the growth chamber that they were in was damaged, and all the gases escaped and oxygen went in, and I lost all of my anaerobe strains, which was a disaster for me. It was I used to teach the anaerobe section for the vet school, and it was a disaster. Plus the awful smell that they produce. Wow, it's quite a memorable problem. I didn't puncture the tent. The janitor was cleaning, and the handle from the mop pierced it, and all hell broke loose after that. It was not a good experience. Air-tolerant anaerobes do not utilize oxygen but can survive and grow to a limited extent. And these are your lactobacilli, streptococcus, and some of the clostridial species will do that. Capnophiles. Now, capno, if you remember, is CO2 in Latin, carbon dioxide. Organisms that grow best at higher CO2s and tension to normal present, and important is the initial isolation of organisms from clinical specimens. Neisseria, gonorrhea, meningitis are the two that causes the STD and meningitis. Brucella causes undulant fever, and then Streptococcus pneumoniae. Believe it or not, we breathe out CO2, so you can imagine that we might see these organisms deeper in the lung because of the higher tensions of the CO2. pH, defined as the degree of acidity or alkalinity. In other words, pH is percent hydrogen, so it's a concentration of the hydrogen ion is the acid, and you go to an alkaline, it gets less and less of that hydrogen concentration, that ion, and then we see more of the hydroxyls up at the alkalinity side. So pH scales expressed from 0 to 14, where 7 is the pH of pure water, because you have equal amounts of hydrogen and equal amounts of hydroxyl groups at pH 7. The pH value decreases towards 0. In other words, the higher the concentration of the hydrogen ions to 100%, which would be 0 pH, 100% of the hydrogen ions, and hydroxyl would be 0 at that point. At the other end of the scale, you would have 0 hydrogen ions, but you would have 100% negative charged OH hydroxyl groups at the alkalinity side of pH 14. So majority of organisms like to grow somewhere between 6 and 7. Now our pH is preferred at 7.4. Now these acidophiles, organisms that thrive in acidic environments, Euglena will do this, and they grow in acid pools. Thermoplasma grow in coal piles, pH 1 to 2. Picrophilus, pH is 0.7, can even grow at pH of pure hydrogen concentration, which is amazing. Many molds and yeast can tolerate acids, common spoilage of agents of pickled foods. So it's all about that environment. The alkalinophiles, which organisms that thrive in an alkaline condition, are Natromonas, live in hot pools, pH of 12. Proteus, vulgaris, and the like, we see a lot of those in sewage treatment plants, can create alkaline conditions to neutralize urine and colonize an infected urinary tract. So Proteus vulgaris and some of these other ones, they have some characteristics that are really kind of nasty. Anything that kind of changes and creates an alkaline type, they call it putrefaction. Osmophiles live in habitats that have higher solute concentration, so it's an osmotic deal, osmotic pressure. If they have a lot of or high solute concentration, that means that it's going to tend for the organism to lose its water. Halophiles prefer high concentrations of salt. Obligate halophiles or halobacterium and halococcus grow at solution 25% sodium chloride, but need at least 9%. And so it's how they set up in transporting the amount of water and salt and all that. Facultative halophiles, remarkably resistant to salt, even though that they don't normally reside in high salt environments. Staph, as we know, can grow on salt, because we looked at that with some of the growth media that we dealt with that had 9.5% salt, and they were able to grow, so it's well within that range. Radiation, phototrophs use visible light rays as energy source. Non-photosynthetic microbes tend to be damaged by the toxic oxygen products. Things oxidize in the sun. Some microbial species produce yellow carotenoid pigments to absorb and dismantle toxic oxygen. You can believe that through millions of years of evolution. UV and ionizing radiation can be used for microbial control, which is a good way. And we've talked about how hospitals use those roving UV light sources to kill bacteria in those, or anything with DNA really, in those facilities. Thermophiles exist in pressures that range from a few times to 1,000 times pressures of the atmosphere. Now, if you've ever been scuba diving, is it 20 feet is one atmosphere, so if you swim down 20 feet you can experience at least one atmosphere, and you go down more than that. Now, in scuba diving, of course, you have to be careful because you compress gases so much as you go back up to get out of the water, that oxygen will expand and you can get lung damage from that. These bacteria are so strictly adapted to high pressures, they will rupture when exposed to normal atmospheric pressure. And it's what I'm saying is, if you scuba dive, you have to stop in your dive at a certain, like 30 feet or 90 feet down, and decompress for a while, and then go up where things tend to equalize. Otherwise, it'll rip tissue, and the same with the bacteria, it'll rip the membrane apart. Gas is trying to escape. The rarest instances, microbes live in shared habitats, association between similar and dissimilar types, and a lot of times these relationships, they can try to help each other by providing some lower the pH because they produce acids around it and that sort of thing. It's all part of a biofilm. That's what they're trying to say here. Symbiosis is a general term to denote the situation in which two organisms live together. These can be mutualism, where it's a win-win, a beneficial relationship. Commensalism, the partner called the commensal receives benefits while its partner, so it's a win and the other partner doesn't really care. And then parasitism is a relationship with it's a win-lose scenario where the host usually loses and whatever's trying to grow on it is winning. And those terms you should already know. Antagonism is association between free-living species that arise when members of a community compete. Antibiosis is the introduction of inhibitory compounds such as antibiotics into the surrounding environment. The first microbe that has a competitive advantage by increasing the space and nutrients available to it. And common in the soil where mixed communities compete for space and food. So these are just competitive type of environments that we see with microbes. And we know this in terms of the gut. We talk about probiotics and the like. And it is an antagonistic type of environment where they try to defend a particular region for themselves. Synergism is an interrelationship between two organisms that benefit them, but not necessarily for survival. Together, the participants cooperate to produce a result that neither of them could do alone. Gum disease, dental caries, what we're talking about, of course, are biofilms. And we see this in various places. We've already talked about that. So the epitome of synergy is a biofilm. In other words, we usually have a selective number at first. The pioneer attaches to a surface. Then other microbes attach to those bacteria and start producing other substances that other microbes can attach to, and as it goes for a biofilm. And you can review the diagram in your book. It talks about the this formation. This is very, very common. We can see this occurring in, unfortunately, and sometimes in replaced hips or dental caries, tooth decay, that sort of thing. And we see the biofilms, but it's more and more of an interest for a lot of scientists. And so it's all about biofilms. Bacteria biofilms behave and respond very differently than free living. When they interact, they activate certain genes that turn on certain characteristics which normally they would never express. The last part, we're going to talk about the growth characteristics of bacteria, and this is really straightforward, but again deals with a lot of terminology. Binary fission, where one cell becomes two, duplicates its chromosome. There is no sex of male, female, or any of that, and about every 20 minutes it just separates, and the main concern is duplicating that huge circular chromosome, and it does that very effectively. So you can see one cell starting to replicate its chromosome, equally distributes it, and then you've now got two cells. That's binary fission. The generation time or doubling time is the time required to complete the fission cycle, binary fission. A generation increases the population by a factor of 2. So, we know that in our own families to having a generation. As long as the environment remains favorable, the doubling effect will continue at a constant rate. So if it doubles every 20 minutes and so forth, it will. The length of the generation time is a measure of the growth rate of the organism. The average generation time is 30 to 60 minutes. There are some that can grow 10 to 12 minutes, and most of the staph and strep that we see, we know it's probably in the 20s and 30s. Mycobacterium leprae, or leprae, has a generation time of 10 to 30 days. It's really difficult for this organism to compete with the high iron acquisition of our own body, so their growth rates grossly short. It takes them a long time to grow. Environmental bacteria have generation times measured in months. Most pathogens have a relatively short generation time because it's a smorgasbord or a salad bar, essentially, once they get in. So the mathematics behind the population growth is really, when you think of bacteria, it's exponential. In other words, when one divides, you have 2, and then those 2 now have 2, and it multiplies as it goes across. And there's certain ways that we deal with this sort of math, and we look at the size of the population. I'm not going to hold you to knowing this equation, but just know that the growth curves of these depend on being able to count the number of bacteria, and finding out their growth rate is important sometimes in pathogenesis. So if you were to wait a certain period of time, like every 60 minutes, and then take a sample out of the growth, you could see that the number of microorganisms grow exponentially as we go across. So logarithmic is usually what we use in terms of logs. Logs are base 10, so you can see we see almost a linear type of an association, where a linear growth shown just plotted straight out without any differences in the lines. We see this sort of a curve, rather pronounced curve there, where a logarithmic, these you can't see, the demarcations aren't there, but the spacing between 10 and to 100, it starts to change logarithmically, so it spreads out unevenly across, so that's why you get this linear type of line in your output. But that's linear. As far as the logarithmic output is log base 10. So here's those lines I was talking about, and you can always identify those as logarithmic, and it helps us plot to see a trend and that sort of thing, and measure rates easier than you can with these straight plots, linear scales. So logarithmic scales usually are base 10, and you can see as we go from 10 to the 8th, 10 to the 9th, you can see the line spacing changes as we get closer to the next number. That's logarithmic. So if we want to represent, and let's say using an optical density type of thing and measuring absorbance, you can see the linear nature. We always plot those in a logarithmic type of arrangement. The lag phase is a flat period of growth due to newly inoculated cells. This is a problem when you're trying to scale. I used to work and we grew things in fermentation vats, really large ones, and you inoculate them, and it may be almost 12 hours before you start to see any really particular growth happening. We call that the lag phase. It takes a while for the bacteria to sort of to adjust and develop their enzymes and all that. Exponential growth, or the log phase, is the growth curve as they're zooming. They're growing at full bore speeds, and that's the exponential growth. And now they start to run out of media or some of the certain characteristics. We call this the stationary growth phase, where the birth and the death kind of are equal, and then it slows down into the death phase, and that's where you start to see the bacteria dying, and then you see viable non-culturable state, which is the VNC, which just means that there may be cell debris there, but they're not, we can't grow them, so the viable cell count is greatly reduced. So here's the lag phase, exponential growth phase, and it starts to run out of particular things in the media, and then it stays stationary as long as it can. Actually, they have no shame. They'll eat each other, and they don't care if things have been used. They'll eat it again, and then eventually the pH and all that things start to change, and then we go into this death spiral, and it's usually the end of the culture. The tendency of populations to exhibit phases of rapid, slow growth and death has implications for food, of course. The growth phase are more vulnerable to antimicrobial agents, so they have to be active. Now, sometimes we might see microorganisms, even though you put antibiotics on your patients and all that, if the microorganisms is sort of static mode, those antibiotics aren't going to do anything. And so a lot of time we have to be concerned about actively growing cells are more susceptible to these sorts of antibiotics. Persons actively shedding bacteria in the early middle stages of infection are more likely to spread than a person in later stages. When we're analyzing culturing, we look at the turbidity, and what that means is in a liquid culture. When you see the word turbid, that means immediately we're talking about a liquid, and then how many cells are there. It turns the transparency, or what appears to be denser and denser, and then you start to see this more turbid type growth. And you can see the difference here. You can see text and things behind it on the ruler, and then if it gets more turbid, now you can't read through it anymore, and it's just actual cells, bacteria cells. You can imagine the number, 10 to the 10th, 10 to the 11th, huge number of bacteria. I bet you that would be tasty. Not. So representing the growth rate, the more turbid it would be up there, and that's why the numbers would change on the spectrophotometer. Now we can count cells in various ways. We can do a direct cell count by just plating the bacteria out, which is a pain. We can use a coulter counter, which scans. In other words, every time it passes through a particular point, it flashes the light or blocks the light, and that pulse can be counted. Flow cytometers work in a similar way, but they can even measure the cell size, so it's more detailed. So these are interesting ways of counting bacteria. I've seen flow cytometers. They're used pretty frequently. And then we can use probe PCR and quantify by the amount of DNA that we can obtain back. This is in my era. We used to use these little sort of microscopic counters, and you pick out a square, and then we know how many squares there are on a particular slide for the bacteria, and then we can just multiply that by the number of cells that are in that square. Let's say there's 10, and let's say there's 30 squares in your plate, and there's 300 bacteria. So it's a count that you can use without actually counting all of them. There's a coulter particle counter. It's really simple. You just kind of put your sample through, and it looks for the impedance. So you can see electrically as something passes through, it's sort of like interference in the magnetic field, and you can detect it in a voltage drop. And it's widely used as a counter. Flow cytometry is that we actually cell by cell passes through. The laser goes through, and either it is not blocked or is blocked, and every time it is blocked it generates a pulse which we can count using a computer, and it gives you really accurate type of counts this way. We use this a lot in the pharmaceutical industry. Real-time PCR is polymerase chain reaction. I won't go through the details, but the key to PCR is it amplifies DNA. What DNA? These are primers that are specific for a particular region of DNA. So the only thing that gets copied is that region in between the two primers, and we can make lots of copies of those, and that's PCR. If you have a lot of the original DNA, then obviously you'll get more DNA faster. So it's the amount of time and the numbers of how much DNA you can acquire will give you roughly the original count. It's easy to do, and you can run it in about 3 or 4 hours, so that's why it's there. Put the steps of the growth curves in correct order, and I'll give you a few seconds. Very good. We go from the lag phase to the exponential phase, then we go to the stationary phase, and then we go into the death phase. So that's all we have for this particular chapter. We've done it for this week, and we'll be moving on to the next prescribed chapter. And so this is chapter 6, and then we'll be moving on to the next one, and you'll be looking for that. I'll be posting it perhaps over the weekend or so. Great to have you. I hope you're doing well. And I sent around a poll just to get feedback on these types of presentations. Let me know what I can do better. And then we start facilitating more within the teams. So read about or watch the videos about how to use Teams. I'm going to provide a case study next week, and in Teams we can work together on tearing those apart. And it's fun. Have an open mind about it. All right, see you next time.